After Mr. Burger enters the mouth, it is chewed up and becomes a bolus. Then the starch gets digested there with the help of amylase, an enzyme that's secreted by the salivary gland. The food that has been partially digested by amylase then gets chewed up. The saliva that also contains mucus, which lubricates the food, and also contains hydrogen carbonate, which provides the ideal pH level for amylase to work. After Mr. Burger is chewed and starch is digested, it then passes down to the esophagus. Mr. Bolus then quickly moves down the esophagus to the stomach in one continuous peristaltic wave. The purpose of the muscle contraction, called peristalsis, is that it forces food to travel downward and prevents backflow of food. This is useful when bodies are turned upside down when swallowing, as the muscles do not take into account of gravity and still allows the partially digested food to pass down to the stomach successfully. There are two circular muscles in the stomach that control the backflow of food: cardiac sphincter and pyloric sphincter. These muscles are vital to the stomach organ because they prevent acid backflow so that the lining of the stomach does not get damaged and form ulcers. The gastric gland secretes hydrochloric acid and pepsinogen, which is the inactive form of pepsin. The pepsinogen is activated by HCl, and then they combine to form gastric acid that digests proteins into short peptides.
And now comes Mr. Kime from the stomach for more work to be done. As the food passes the small intestine, most of the chemical digestions take place. Small intestine is made up of three parts: duodenum, jejunum, and ileum. This is done through a muscle contraction called peristalsis. Peristalsis is separated into three steps. First, there is a contraction of circular muscle which binds to Mr. Kime. Then another contraction takes place using longitudinal muscle ahead of Mr. Kime. Lastly, there is a contraction of circular muscle with each layer created forcing Mr. Kime to move forward. Food is broken down by enzymes from pancreas and bile from the liver as it heads towards the lumen to carry hydrolysis. For nutrients to pass from the lumen to capillaries or lacteals in the villi, it must go through villous epithelium. This process of absorption can be seen in duodenum, the first part of intestine, where most of the absorption takes place. The first thing nutrients meet are the villi, which are located on the wall of the small intestine, and their tiny elongated projections help them increase the surface area for the absorption of nutrients and other substances from small intestine. These villi are capable of absorbing monomers formed by digestion, mineral ions, and vitamins. As nutrients enter the absorption phase, triglycerides are digested to fatty acids and monoglyceride. While monoglyceride can pass into the villous epithelium by simple diffusion, fatty acids are absorbed by facilitated diffusion by fatty acid transporters. When they enter villous epithelium, fatty acids are put together with monoglycerides producing triglycerides. This blocks them from returning to the lumen of small intestine. Triglycerides then get coated with phospholipids and protein in order to exit the villous epithelium into the interior of villus as lipoproteins.
Sodium-potassium pumps in the inwards-facing part of the plasma membrane pump sodium ions by active transport from the cytoplasm to the villus, and potassium ions in the opposite direction. This helps to create a low concentration of sodium ions inside the villus epithelium cells. Sodium-glucose cotransporter proteins in the microvilli transfer a sodium ion and a glucose molecule together from the intestinal lumen to cytoplasm of the epithelium cells. This facilitated diffusion is passive, but it depends on the concentration gradient of sodium ions created by active transport. Glucose channels allow the glucose to move by facilitated diffusion from the cytoplasm to the interstitial space inside the villus and on into blood capillary in the villus.
Pancreas enzymes are secreted into the lumen of the small intestine. In order to do so, the small groups of cells secrete hormones, insulin, and glucagon into the blood. Others synthesize and secrete digestive enzymes. This is all capable due to hormones being synthesized and secreted by stomach and enteric nervous system. The digestive enzymes that were secreted are then synthesized in pancreatic gland cells on ribosomes of Raphier. Then they travel into Golgi apparatus where they are secreted by exocytosis. The ducts of pancreas begin to merge one by one, forming one pancreatic duct. A liter of pancreatic juice, which contains enzymes that digest amylase, lipase, and protease, is secreted into the lumen of the small intestine every day.
Liver plays multiple roles in the digestive system, but its main responsibility is to process the nutrients moving in from the small intestine. Bile from the liver is secreted into the small intestine, and the secretion of surfactants in bile help to break up lipid droplets. Simply put, liver is the human body's chemical factory. It turns the raw materials from the small intestine and produces various chemicals the body needs to function properly. Another important role that the liver plays is detoxification of harmful chemicals, such as breaking down and secreting unneeded drugs. Gallbladder is found inferior and posterior to the liver. Even though the size of it is relatively small compared to others, it plays a very important role in digestion processes. Bile produced from the liver is held in the gallbladder until a time comes where fatty foods need to be digested in the duodenum of the small intestine.
Large intestine is a long muscular tube of 6 feet length. It acts as a connecting point between the small intestine and the rectum, which leads to the anus. There are 4 parts to large intestine which should be noted: ascending colon, transverse colon, descending colon, and sigmoid colon, which connects to the rectum. When food passes by large intestine, it is responsible for processing waste. Waste, also known as stool from the digestive process, is passed through the intestine through peristalsis, turning from liquid state to solid. After 36 hours of constant movement, the waste gets through the large intestine, leaving food debris and bacteria. These bacteria play an important role as they protect our body from any harmful bacteria. As the stool or feces pile up, it is ready to move on to the rectum and anus for elimination process. Finally, at the last step of digesting Mr. Chaeyeong, anus has a structure of 2-inch long canal with pelvic floor muscles and 2 anal sphincters. When the upper anus detects rectal content, the body can tell whether the content is liquid, gas, or solid. The sphincter muscles around the anus controls the entrance of the stool, while pelvic floor muscle creates a certain angle between the rectum and the anus, controlling the amount of stool that goes through. Internal sphincter is always kept tight, except for when stool is entering. The external sphincter holds the stool until it is able to be eliminated. Out goes Mr. Poop, and somewhat different from what Mr. Burger looked like.